Hi everyone, my name is Jillian Nooner and I'm a rising senior majoring in chemistry in the college. Today I will be presenting on my research in the Eisenlohr lab this summer. Specifically, my research focuses on Ectromelia virus, also known as mousepox, which is a pathogen within the orthopox virus genus. C15 is a member of the B22 family of proteins within the orthopox virus genome and has been shown to be necessary for the lethality of Ectromelia virus in mice. This indicates to us that C15 is a virulence factor in Ectromelia infection. A lot of previous work dealing with C15 has shown its ability to inhibit both CD4 T cells and the degranulation of NK cells, making it a very unique protein and that it's able to evade both the adaptive and the innate immune systems. Despite this knowledge, relatively little is known about the ways C15 operates at the molecular level. And a lot of my research this summer has been devoted to developing tools and optimizing protocols to help us study this protein. I began my project by developing a stable cell line that simultaneously expresses the mouse MHC2 molecule, its transactivator, and the HA tag C15 protein. I created this cell line using the Sleeping Beauty transposon system, which is depicted in Figure 2, and also incorporated a TET-on system to allow for the inducible expression of C15 when incubated with doxycycline overnight. Using the cell line, I was able to design and run an immunoprecipitation protocol to pull down C15 and hopefully identify its binding partners. I performed this immunoprecipitation with an HA-specific kit. I then took the eluates and ran a Western blot with an antibody against HA. In Figure 5, we can see that C15 is present in the fourth lane, the elution lane, which indicates that this protocol is successful in isolating the C15 proteins from my cells. I then took this a step further and performed the immunoprecipitation a second time, but I stained with anti-mouse MHC2 in my Western blot in an attempt to detect components of the antigen presentation machinery. I performed the Western blot twice, and each time we observed a very faint band around 28 kilodaltons. However, we want to perform an MHC2 immunoprecipitation followed by an immunoblot for HA in order to determine whether these bands are actually significant. And down the road, we also want to run this immunoprecipitation protocol, isolate the elution, and run mass spectrometry to more quantitatively determine the molecular binding partners of C15. The last aspect of my project focused on generating a recombinant Ectromelia virus that included only the N-terminus of C15, which roughly corresponds to amino acids 1 through 456. I did this by first creating a plasmid that contained the N-terminus of C15, as well as a V5 tag. I then took BSC1 cells that had been infected with Ectromelia C15 knockout, a viral construct in which the C15 coding region has been replaced by GFP. I transfected these cells with my plasmid in order to promote the desired recombination event. However, it's important to note that this recombination event occurs at a very low frequency. So I utilized a CRISPR-Cas9 selection system in which I exposed the recombinant product to GFP single guide RNA. And because poxviruses do not have DNA repair machinery, exposure to this selection method and the single guide RNA initiates abortive replication. Therefore, any viruses which do not undergo recombination will still contain GFP and be selected against in this method. We ran 3 rounds of this selection and isolated plaques that presumably contained our recombinant virus of interest. We then lysed these cells and ran a Western blot, staining against both V5 and HSP70 as our control. In Figure 7, we observed a band corresponding to the length of the N-terminus in the condition that produced our desired recombinant virus, which leads us to believe that the selection method was successful and allowed us to generate this N-terminus viral construct. This will be incredibly helpful going forward because it will allow us to examine whether the N-terminus is necessary for T cell inhibition in vitro, and we can also look at how it impacts NK cell function when it infects mice in vivo. I learned a lot this summer, and I'm very excited to continue working on this project throughout the year. I want to acknowledge PennCurve for funding my project, as well as the Eisenlohr Lab for providing me with many opportunities to learn this summer. Thank you.